Okay, so we have gathered here to talk about the most important advances in lymphoma over the past 5 years. I'm sitting here with a panel of colleagues who work in different aspects of the disease. My name is Ari Melnick. I am a hematology oncologist from Weill Cornell Medicine in New York City.
I am Elias Campo, a pathologist at the University Hospital of Barcelona.
I'm Reinhardt, a hematologist from Cologne University in Germany.
I'm Jude Fitzgibbon. I'm a research scientist at Queen Mary University of London.
Riccardo Dalla-Favera. I work on lymphoma pathogenesis at Columbia University, New York.
Great. So the last 5 years have really been, I think, incredible for us in terms of the speed and number of advances that have occurred. It's almost hard to list them in some kind of priority order because there are so many of them. But certainly one of the ones that we can all agree on is the fact that we now for the first time understand the genetics of the disease. We have for the first time very thorough maps of the mutations that occur in lymphoma. And that has certainly been a transformative event because it points now to the mechanisms that drive the disease and therapeutic targets that could be taken advantage of to improve the treatment of patients in a more precise way. In many ways, these mutations also give us the ability to develop model systems to understand how lymphomas develop from the beginning. And so maybe I thought, Riccardo, you can give us some thoughts about modeling lymphomas and how the mutations have changed our ability to understand the pathogenesis.
Modeling in mice, maybe?
Yeah.
So I think the knowledge that Ari has described about the coding genome of lymphoma, the full map of genetic lesion, allows us to test in the usual in vitro system, but more importantly, creating mouse models in which we can ask specific question on what is the role of each individual genetic lesion in the overall construction of a lymphoma disease. I think the mice have given a lot of answers in terms of, again, the individual role, and also represents, in some cases, a very useful tool for preclinical testing of drugs targeting the various mechanisms that we have identified. I think they're used for research, they've been used for the industry, also for testing new drugs before they get into the clinical arena.
You have a lot of experience also developing mouse models.
Yeah, I can only echo what you just said. I think it's great because it gives you confidence that you are modeling the right thing if you follow what has been described in these genomics papers. I think there's much more to these genomics papers than just allowing us to create better models for preclinical research. I think what it also may lead us to is to more precisely designed trials. Right? We have these subgroups now that are genetically defined. We can relatively easily, I think, develop sequencing panels to distill these patients harboring certain mutations, which will then hopefully allow for more precise trials with less patients needed to receive clinical signals. I think there's really broad possibilities ahead of us for the next couple of years.
One point that Christian alluded to just now is the new ways of classifying tumors, lymphomas, based on their genetic composition. I'm wondering if you could expand on that a bit and explain how that's potentially transformative.
Well, I think the exciting thing really is that probably for the last 20 or 25 years, you could say that several institutions around the world have been collecting material very rigorously in relationship to different entities. And probably, Elias, you've been seeing kind of under the microscope different entities from a pathological point of view, but not really having the tools available to dissect out what exactly is the heterogeneity that you're seeing at the molecular level. I think what we have today is the opportunity to kind of get a full personality of cancer based on looking at individual gene mutation and gene expression and a multitude of other techniques that are now coming on stream, like single cell analysis, whereby we can, rather than seeing a single entity under the microscope, so for example diffuse large B-cell lymphoma, we now know for the last several years that there's at least two different types, the ABC type and the germinal center B-cell type, and we can define very rigorously the genetic components, the gene expression changes that dichotomize those groups. And I think there's a sense in us all that rather than taking each entity as a single disease, that if we can subgroup or subdivide patients into different groups, we might be able to personalize treatment in some fashion that would actually increase the overall survival in certain cancers where outcomes are still quite poor. But I think there's a sense that collecting tissue with allied clinical information and then having the opportunity to rigorously dissect the genetics of those tumors is really informing work that Christian is doing, that Riccardo is doing, and Ari, and building a kind of this full personality of the disease.
Yes, it's been encouraging to see multiple groups proposing, publishing different ways to classify lymphomas more rigorously based on the genetic mutations that have biological implications as well for the way the tumors might work and how they might be treated. I'm wondering from the diagnostic perspective how you think the diagnosis of lymphoma has been transformed or changed either by these genetic tools or other means that have come up in the last couple of years.
I think definitely the large amount of information that has been generated in the last years because of the massive sequencing of not all, but a large number of entities has provided us with information that is useful in different ways. One is conceptually. Some of these mutations are allowing us to delineate some entities that were poorly defined. For instance, I am thinking in one particular entity that was considered close to Burkitt's in pediatric population, but then when we analyze the profile of mutations, clearly the profile was totally different from Burkitt's and really closer to germinal center DLBCL. So by analyzing the profile of these mutations conceptually help us to situate the entities in the right conceptual framework. But also there is a great impact in the daily practice. There is now different mutations that came out from these genomic studies that we are now currently using in the differential diagnosis of some entities. We know for a long time that there is not a single mutation that defines an entity. Most of them are shared by different lymphoma entities. These mutations in the context of other morphological phenotypic information that help us to delineate the entity. But certainly there are borders that are difficult to define with the conventional approach that the pathologists had. And this genetic information is definitely being very useful to make a more precise diagnosis. So I think that's one of the major changes in the pathology practice in the last years.
So one of the concepts that's emerged in oncology in the past 5 years is precision medicine, which is defined loosely as the ability to diagnose the molecular basis of a given individual's tumor and assign them to therapy based on those molecular features. And the molecular feature that is most commonly captured in tumors are the mutation spectrum. So precision therapy winds up meaning mutation-targeted therapy for patients based on their genetics. I'm wondering how you feel about that concept's relevance to lymphoma and whether or not it's something that is able to be translated into the clinic at this stage. How close or far are we based on the state of the diagnostic methods that you can apply?
I think that's the way we all think we are going to go. There is a better understanding of the mechanisms that are leading to the development of these diseases. Definitely, we all think that targeting these key mechanisms has to be an important way to stop or including cure the disease. We have some examples, for instance, that we always mention the success with chronic myeloid leukemia, Unfortunately, we cannot reproduce this model in many others, but definitely this is the way to go. However, on the other hand, I think we have to be careful because the biology of these tumors is more complex, and not the same mutations in the same context mean the same. I guess, yes, acute lymphoblastic leukemia T-cell type might carry notch mutations, and CLL also carry notch mutations. Should we treat notch mutations, or we should treat CLL and we should treat TALL? So I think this context of genetic alterations driving therapy is very important, but I think that has to be put in the right context.
And I think you've made a very good point about the notion that it's complex, because I think that the tumour cell must be co-opting several different pathways. And I think up to now, the only way that we can actually approach clinical trials is to take a single pathway at a time and target it. But in actuality, we need to be thinking that the tumour is complex, and we need to be, in the future, able to hit maybe 2 or 3 different nodes, and hopefully the combination will actually have a greater effect.
When it comes to precision medicine, and one other thing is important too. I mean, we have tools now to also genetically dissect vulnerabilities in tumors and lymphomas. So with the advent of CRISPR-Cas technology, we can really go on a single gene level and see whether we find vulnerabilities in the context of a certain tumor genome or lymphoma genomes. I think that is something that will probably drive the design of our future trials and also drug discovery, because we may make unexpected discoveries using these technologies. So I think combining the models with CRISPR screening has great potential also to inform then patient treatment.
So Riccardo, you were the first to discover a mutation in lymphoma. So you're kind of like the father of this field.
A long time ago.
Yes. But I'm wondering, with your view over all these years of how that's evolved, how do you view this?
Well, the history was, everything started with the molecular biology explosion in the late 70s, when you can start cloning genes, right? And as you know, the first indication were recurrent chromosomal translocations that led us to identify the specific players that were in this cytogenetically microscopically visible lesion. For I think 20 years, the search of mutant genes was actually focusing on educated guesses, cytogenetic input, and so forth. Everything changed with next generation sequencing that led to the current knowledge of the enormous repertoire of mutation. I think we are just maybe halfway before the whole the story is complete. In many ways, the organization of the enormous knowledge that we have of mutation in completely understood biology is incomplete. The knowledge of what goes on in the genome is also incomplete. We know almost everything about the coding genome, which is what 3-4% of the whole genome. There's going to be certainly a lot coming out of mutations in regulatory domains in different lymphoid diseases. And ultimately, at least my hope would be that this enormous repertoire of genetic lesion will be restricted to a much simpler and more manageable pathway catalog that will allow to develop targeted therapies that probably will take advantage, target different genetic lesion, all make the same alteration in the same pathway. So I think in the next years we'll see still a lot toward real precision medicine.